another topic, and that is immunological tolerance. A little bit of background. Well, first of all, the definition of tolerance. Well, it is a poor or lack of response to a particular antigen, very specifically to that particular antigen. Unresponsiveness, when you give cyclosporin or anti-lymphocyte serum or anti-CD3 antibodies or anti-CD25 antibodies in the case of transplants or other situations, or you use methotrexate, melphalan, Cytoxan, those are all very broad immunosuppressive agents. They suppress an immune response to any antigen, every antigen. Okay? Tolerance refers to an antigen-induced specific unresponsiveness. The best example is we are tolerant to all our tissues. Okay? I'm a B positive. Okay? I don't respond to the B blood group antigen, but the antigen is there as an immunogen in another person, if they happen to be O or A. Okay? So we are normally tolerant to all our components. We may produce a small amount of reactions to some proteins or some antigens, but they are not very large. So tolerance does not mean total lack of responsiveness either. It may be undetectable. Okay? Now, a little bit of background history. The experimental or the mechanism of tolerance were obviously a mystery, and it is still not fully defined, but they will talk about what are the components of those mechanisms. In the 50s, it was observed that bovine fetus is different from humans. If they have twins and they are dizygotic, they have a common circulation. Therefore, there's an exchange of blood from each other. And when they are born, they don't respond to each other's blood products or any of those things. And that was the observation in the 50s. And then there was a group of scientists, one American and two British in England. The American scientist from Texas was visiting postdoctoral fellow in London, and they designed an experiment. And the experiment consisted of this. They took newborn inbred strains of mice, we'll call them A. All of the A mice are identical twins, or identical whatever number you can think of, but they are all identical. And injected to them either syngeneic or isologous, you can say, bone marrow at birth, or homologous, allogeneic, from strain B. This is bone marrow. In mice, fortunately for them, there are not very many T cells, mature T cells, to cause graft-versus-host disease. There are some casualties in those experiments, but the surviving, a significant number of animals survive. They left them for about 8 weeks to become young adults. Mice become young adults in 8 weeks' time. And then they grafted them with either the two bone marrow type skins. In other words, type B, strain B skin, or strain A skin, or even a third party, strain C skin, as you can see here. Black and amber, and then there is another third-party skin there. Okay, and then left them for another 10 days and observed over that 10 days period, and they found that the animals that were given strain A, they rejected both grafts, the type C and type B, while they accepted, of course, as would be expected, their own genotype skin. Amazingly, also, the mice that were given the bone marrow from B strain, they also accepted the B as well as A strain graft, while rejected the C type, a third party. So the conclusion from that, they had induced tolerance in these animals by injecting them with foreign cells at birth, and that tolerance was very specific to that particular type of tissue antigens, because they were still responsive to a third party, a third type skin. And those observations were published in the early 60s. As a matter of fact, that made a basis, formed a basis of Nobel Prize to these three scientists. And the picture you saw on the last slide was one of the group leaders, Peter Medawar, Sir Peter Medawar. He got knighthood for that after that too. Okay? So, and here's a picture of that white mouse being given bone marrow cells at birth from a black mouse and becoming tolerant, and there's a skin graft accepted there. Okay, so that's the basis for immunology. What does one conclude from that? That there is a certain time at which you can expose an individual to foreign antigens, and they become tolerant. What are the major factors that determine whether that an individual will produce an immune response or tolerance are listed here? Physical form of the antigen is important. Dr. Mayer told you about it, mentioned to you that physical form of the antigen, if it is particulate antigen, complex antigen, particulate or complex antigen, it favors an immune response if they are large, aggregated, complex molecules. On the other hand, if they are simpler molecules, soluble molecules, aggregate-free, they tend to produce unresponsiveness. And the degree of unresponsiveness will be different for each molecule. Okay, antigen processing, if they are properly processed and presented, they will produce an immune response. If they are improperly processed or improperly presented, then they will produce immunological unresponsiveness. And we'll give you some examples of these later on. Okay, route of injection. He did tell you that while subcutaneous, intramuscular, those are the routes that are normally used for immunization, are effective in producing a good response. Oral immunization, sometimes even intravenous inoculations, tend to produce unresponsiveness. Dose of antigen. There is a threshold of antigen dose. Very, very small quantities of antigen produce unresponsiveness. Very large amounts will produce also unresponsiveness. There is an optimal dose, and that can vary for each antigen. An optimal dose that will be good for immunization. So very large and very small doses produce unresponsiveness. Okay, some more factors. Age of the responding animals. You saw in that experiment. At birth, when they were treated with foreign cells, they became unresponsive. And that was not later on, it was discovered for some experiments, it was necessary to give a thymic graft at a very early stage, thymic graft even, cause immunological unresponsiveness or tolerance. So any exposure at very early age, that's why if you looked at that chart for different antigens. There was different optimal age. That was by experience that the schedule had this antigen is good, effective at this particular age and not at this particular age. You may remember a few years ago there was an epidemic of measles, I think, or measles or rubella as a matter of fact, it interrupted some of the basketball games as well. One of the basketball superstars also came down with the disease. And what was discovered is that they were immunized at a very, very early age. And after that, there was a directive from the CDC that all those children that were immunized at that very early age should be given boosters, okay? Additional boosters. And the age of first immunization was changed to 1 year or thereabouts. Okay? So different antigens, if their individual is exposed too early, it may produce unresponsiveness. Adults and immunologically mature animals, humans, they are good responders, where a newborn, in the case of mice, you saw it particularly, are immunologically immature, whatever reason it may be, they will produce unresponsiveness. Okay, differentiation of status, that maturation is at the cellular level as well. If differentiating, not fully, if one exposes the cells that are fully differentiated, or they are memory cells, they'll produce a good response. However, if they are undifferentiated, like in B cells or with only IgM on their surface, if you remember sequence of immunoglobulin expression on T cells and on B cells, immunoglobulin IgM comes up first. Those are not fully differentiated. If such cells are exposed to antigen, they produce unresponsiveness. Same way T cells. If they are not fully differentiated, not fully mature, and they're exposed to antigen, they become tolerant. If you remember Dr. McCaleb's lecture, that's the basis for negative selection. Cells that are just in the process of differentiation, they have got very low levels of TCR on their surface. They interact with MHC presenting self-antigens and they become unresponsive. Okay? So if they are not fully differentiated cells, they become unresponsive. Okay? Some definitions of tolerance, some characteristics of tolerance. It is antigen-induced and an active process. In other words, there is a response in the body. In the process of negative selection, that is a response. They recognize the antigen and they is a signal given to the cell and they undergo apoptosis, programmed cell death. Okay? So it's an active process, it's antigen-induced, just like an immune response. So there's a lot of similarity between immune response and induction of tolerance. Like immunologic memory, it is antigen-specific. Okay? And like immunologic memory, it can exist in B cells, or T cells, or both, depending on what determinants on that molecule are producing that tolerogenic response. Okay, like immunologic memory, it is easier to induce and lasts longer in T cells than B cells. If you remember Dr. Mayer, when he was talking about responses, memory T cells, he told you that memory T cells are a lot longer lived. As a matter of fact, some memory cells have been traced about 40 years. Okay? So they are, likewise, immunological memory also lasts longer in B cells and T cells. Okay? Mechanisms. What are the different mechanisms that are responsible for immunologic tolerance? Negative selection in the thymus. Well, negative selection does not only take place in the thymus, it also takes place in the bone marrow. As the B cells are differentiating, and there they get, if you remember, Dr. Mayer talked about the heavy chain and then a surrogate light chain being produced, and then it's replaced by the real light chain. Well, and obviously when they start producing those immunoglobulins, they're in a very, very low concentration, and they interact with the self-antigens being at a very, very high concentration. And that's a characteristic environment for clonal deletion of those cells. There's another mechanism that I'll talk about that takes place that is called receptor editing. That's also taking place in the bone marrow environment as well as in the periphery, and we'll talk about that a little bit later. Clonal energy. Lack of the stimulatory molecules. Remember CD28 and B7-1, B7-2, or CD80, 86 from Dr. McAuliffe's lecture. Well, if the B cells, if the antigen-presenting cells, has these molecules and it interacts with CD28, there is going to be a positive immune response. But if it happens not to find that interaction, that co-stimulatory interaction, the cell is going to become tolerant, energetic, non-responsive. And later on, it will die of lack of stimulus. Another molecule, CTLA-4. This B7 molecule, can interact either with the CD28 or CTLA-4. And if it interacts with B7, it's a positive response. If it interacts with CTLA-4, it's going to be negative signal, tolerance, unresponsiveness. Now, receptor editing I mentioned, and I will just talk about it, and then we'll take a break. What happens there is that Dr. Mayer told you that VDJ genes are rearranged, attached to a constant region, gene, is transcribed, translated, that's the receptor. And the cell maintains that receptor for the rest of its life. Absolutely true. In all conditions except one. When there is large amount of antigen and very, very large stimulus to the cell, like we have in our body, large amount of antigens, every antigen is in large quantities. When that B cell or T cells interacts in that environment with the antigen, then there is a reactivation of two genes, RAG1 and RAG2. Remember those two? Absolutely essential for rearrangement of VDJ. They are reactivated in the cell, and their job is to clip that V region gene off and bring in another V region gene, resplice it. And that is what's referred to as receptor editing. And any autoreactive cells that have escaped negative selection in the thymus for T cells or in the bone marrow environment or other environment, negative selection, they can reactivate their RAG1, RAG2 and rearrange, bring in another V region gene to that DJ and go on to have another specificity. It's good for the individual because that another specificity is protective against some other antigen, and also it does not react to the self-antigens anymore. And that's what's referred to as receptor editing. I'll stop there, take any questions, and then we'll continue after the break. Any questions about any of the problems? Yes.